So in the previous lecture, we discussed about target identification. Target identification is a very vast and crucial topic. So there is a series of such videos about the topic target identification, and you can find it in the complete video lecture series of drug discovery and development. So it's all a part of drug discovery and development. You can find it out in the playlist for the same. So target identification. Target identification, let's discuss like in a very summary form what we discussed previously. So target identification is the most crucial step in chemical genetics and drug discovery. It is basically the first and foremost step in drug discovery. So first step is always what? Target identification. It is the process of identifying the direct molecular targets of small molecules such as nucleic acid and proteins. So this is also we have discussed in previous lecture. So the key, okay, now we are talking about the key to a good drug design. The key to a good drug design is working out and capturing the clinical spectrum of the disease. Okay, so first of all, it is working out and capturing the clinical spectrum of the disease. And second of all, based on this, we need to identify or find out the exact role of a potential therapeutic target in the disease condition. So what they're trying to say here is, first of all, you need to find out the full details of the disease, and second of all, you need to find out the exact role of even a potential target, potential therapeutic target in that particular disease condition. So it makes a good drug design. Now, as per Oxford Dictionary of Biochemistry and Molecular Biology, a very good drug target definition is there. I'll be sharing it with you. So they say a biological drug target is nothing but a biological entity. So it is a biological entity that can be or is usually a protein or a gene that interacts with and where activity is modulated by a particular compound. So what they said here is that basically a drug target is nothing but a biological entity. It means it has to do with our body. It has to do with the human body. It can be a protein, it can be a gene. Certain times we have come across drugs which actually alter the genes sometimes, or by altering the genes, basically they treat a genetic disease. And sometimes they alter the proteins. It can be your enzymes, it can be the other biological protein targets in a body which actually interacts and where the activity is modulated by a particular compound. Now this particular compound they are talking about the drug. तो बायोलॉजिकल एंटिटी होती है बेसिकली ड्रग टारगेट. ये ड्रग उस बायोलॉजिकल एंटिटी पे टारगेट करेगा जिसके कारण not only will it interact with the drug, it will also modulate its activity. So it's a very nice definition. I thought of putting it here so for your better understanding. So good drug targets need to be relevant to the disease phenotype and should be amendable to therapeutic modulation. So at least whatever the disease is उसके relation में ही drug target होना चाहिए. Not like कि हम drug like we are talking about heart disease और हमने drug target choose किया as discussed in the previous video COX-2 enzyme. So that should not be the case. Or at the same time you need to have a good therapeutic window to assure that any therapeutic modality aimed at the drug target will not cause side effects. by disrupting the physiological features or functions of the target in healthy tissues. Whatever drug target you choose, make sure that it does not have or does not give side effect to other surrounding healthy tissues or the exact healthy tissue that we are targeting. Okay, so identifying a biological target that is druggable. Okay, we are identifying a biological target that is druggable. So this is a word that you'll also observe in a lot of texts. A target is druggable. Okay, a target is druggable if its एक्टिविटी बोले तो बिहेवियर या फिर अदर टर्म्स में हम बोल सकते हैं कि उसका फंक्शन कैन बी मॉड्यूलेटेड बाय अ थेरेपेटिक ड्रग, कैन बी मॉड्यूलेटेड बाय अ ड्रग. तो ड्रगेबल कब हुआ वो, जब उसकी एक्टिविटी को कोई भी ड्रग मॉड्यूलेट कर देगा. So a question that arises in a lot of people's mind is what makes a good target? How will you know that okay, this is the ideal property of a good target? So few will discuss here. First of all, the target should have a confirmed role. So if a target has a confirmed role in what? In the pathophysiology of the disease, then we say that is a good drug target. When the target expression is not evenly distributed in our body, otherwise there could be potential side effects that we would observe. So the target expression is not evenly distributed throughout the body. Third one being the target's 3D structure is available to assess for better understanding. And the fourth one being that the target is easily assayable, that you can easily conduct a assay on that particular target, enabling high throughput screening. Fifth one is that target possess a promising toxicity profile or potential side effect. So toxicity profile or potential you can have potential adverse effects if a drug acted on that particular target, so that can be predicted easily by using a phenotypic data. So the prediction can be easily done of these things, so then it makes a good drug target. And lastly, the proposed target has a favorable intellectual property status, that makes a good drug target. So many targets are initially identified using some scientific literature or public databases such as drug banks or TTDs. This popular method for identifying drug targets can result in competition amongst drug developers, as like if you take the example of school, when you had school or college projects, if there is a literature that is easily available to a lot of people, it has a public access to it, then a lot of people will choose that particular topic, which in turns will create what? Competition. That oh my god, they are dancing on the same songs, or they are presenting on the same topic. Similarly, if we think it in perspective of pharma or pharmaceutical companies or clinical research, these popular methods for identifying drug targets can result in competition amongst the drug developers. So when it comes to advancing drug discovery process, some are willing to accept more scientific risk to drive forward targets that perhaps have less information available, but also less competition, hopes for driving more established targets forward. So beside literature, there are numerous other ways for drug identification. So the other ways of basically strategies for drug identification, target identification, mainly we'll be talking about the two, which can be based on the two type of target identification strategies. So what are these strategies? First one is target deconvolution, and the next one being target discovery. So target deconvolution, in this scenario, what happens is you begin with a drug that appears efficacious and the target is identified retrospectively. Okay, so हमने क्या किया इस केस में, हमने अभी तक जो हमने बात सुनी थी, that was saying कि first we are identifying the target and then we find our lead compound. But here what is happening is, in this scenario we begin with a drug that appears efficacious. So हम starting किससे करते हैं, एक ऐसे drug से जो कि हमें लग रहा है कि एफिकेशियस है या फिर इसका कोई थेरापेटिक अच्छा इफेक्ट पड़ेगा बॉडी पे, और इसके बाद फिर हम ट्राई करते हैं ड्रग आइडेंटिफिकेशन का, कि अब हम इस ड्रग के बेसिस पे, सपोज आई फाउंड आउट दैट मिनॉक्सीडिल, okay, minoxidil, so this particular drug is helping in blood pressure. So first I identified the drug and then I'll find out कि इसका क्या target होगा. So this is one approach, and the second one is target discovery. So as the word says कि target की discovery कर रहे हैं. So this approach works on the concept that if you want a new drug, you must find a new target. So this is the, like, what do you say, a line that is like a mission or vision. So if you want a new drug, आपके पास कोई drug नहीं है, आपको नया drug चाहिए, then you must find a new target. So once a target is identified, compound libraries are then screened to find a drug that binds to the target and elicits a desired effect. So based on that, we'll draw a very good like a flow chart on whatever we have learned. So this is target identification. ठीक है, अब दो चीज़ होगी टारगेट आइडेंटिफिकेशन में, दो कंडीशंस होगी. पहली है कंडीशन दैट, और दूसरी है कंडीशन. सो पहली कंडीशन है दैट यू हैव अ कंपाउंड, यू ऑलरेडी हैव अ कंपाउंड. एंड सेकेंड कंडीशन है कि यू वॉन्ट अ कंपाउंड, आपके पास है नहीं, आपको चाहिए. सो यू वॉन्ट अ कंपाउंड. नाउ if you have a compound, then the approach of like target deconvolution can be done. But if you don't have, and if you want a compound, then target discovery needs to be approached. Now what will happen is that this is done on the basis of this particular thing. How deconvolution is done? It is done by phenotypic screening approach. And how target discovery is done? It is done by target based screening approach. So in either of the cases, whether it is deconvolution or whether it is discovery, one thing is for sure, that you will find a target. You will find one target, and after this we are going to do the target validation. We'll validate whether the identified target is good or not. So you can take a screenshot of this flow chart if you want. So in principle, there is no regulatory requirement to know the molecular target of a drug or clinical candidate, since all that matters at the end is that a drug is safe and efficacious. In fact, there is like a number of approved drugs for which the MOA is unknown. However, the drug development process is obviously greatly facilitated if the target is known, since this enables the rational design, which we learned in previous lectures, of new molecule with like improved potency and safety profiles. So obviously you'll be more successful if you know the target. So now a little bit on target deconvolution and target discovery. So phenotypic screening, okay, as we learned here. So there is phenotypic screening. Phenotypic screening is a strategy used in drug discovery to identify molecules. We use it to identify What we use it to identify molecules with ability to alter. So they have the ability of doing what? They have the ability of altering. So they have the ability to alter what? Again the question, what? So they have the ability to alter a cell's phenotype. Any similarity between these two? So animal models and cell-based assays are both the strategies used to identify these molecules. So we do phenotypic screening to identify a molecule that has the ability to alter a cell's phenotype um, based on like what? We take the help of animal models or cell-based assays to do so. Now, the phenotypic approach to drug discovery falls within the realm of target deconvolution and involves exposing cells or isolated tissues or animal models to small molecules to determine whether a specific candidate molecule enters or exerts the desired effect, which is observed by a change in the phenotype, which is very quite obvious. Now, the phenotypic approach goes beyond individual proteins or nucleic acid and involves the study of entire signaling pathway. Entire. So, the drug effect is determined before the specific biological drug target is identified that like underlines the observed phenotypic response, and that is identified. So the advantage is that over target based is their ability to demonstrate the efficacy of a drug. So if you have like if you want to draw a parallel, so I can say the advantage to it is that over the drug based discovery is that it has the ability to demonstrate the efficacy of a drug efficacy of a drug in content of a cellular environment. So drug is acting on the target in its normal biological content rather than a purified target in a biochemical screening. So that is the major advantage. Now target discovery we'll discuss a little bit. Target discovery approach, which we discussed here, that target based screening approach. So, biological drug targets are already established. Whatever we were studying in the like before studying the phenotypic approach, whatever in general we learned throughout these lectures is what is the basis of this particular topic. So, these are already established. What are established? The biological targets are already established or discovered before the lead discovery. So, target is already identified or established or discovered, whatever term you want to use, before lead discovery starts, before identifying what a drug substance actually is. So hence the target discovery is cornerstone of target based screening. So the target role in a disease process is known. This target is then used to create relevant system based assays, and vast compound libraries are screened in search of a hit candidate drug. The fact that knowledge of a drug candidate's molecular mechanism is understood. So एक drug candidate होगा हमारा, but उसका जो molecular mechanism है उसी को हमने समझ लिया, we understood it. So drug candidate's molecular mechanism is understood from like offset presence as a key advantage over like phenotypic approaches, and target based methods are typically easier to carry out. They are less expensive to develop and it is a faster process. So these are few of the advantages of drug target based discovery over the phenotypic based target identification. So target is like to summarize it in a way, we can say that target identification lies at the heart of like a modern drug discovery. So whatever the rational drug discovery we discussed in previous lectures. So on paper, the process sounds simple, like find a biological target that plays a role in a disease, and then find a therapeutic drug that interacts with it, and yet it like is a very complex task to do when you actually implement and execute it. So the challenge of drug discovery and validating those targets is reflected in the failure rate of drug candidates in the clinic. So when potentially at the end हमने सब कुछ आइडेंटिफाई कर लिया, लास्ट में हमें पता चल रहा है कि दिस ड्रग का एक्चुअली इतनी एफिकेसी नहीं है, या दिस पर्टिकुलर ड्रग टारगेट पोटेंशियल ड्रग टारगेट के ये साइड इफेक्ट्स आ ही रहे हैं. So rather than finding out in the later stages when a lot of time and resources have been spent, we are getting to know it beforehand. And this is like a way by which promising treatments को fail होने से हम बचा सकते हैं. So the reason for such failures is usually that underlying hypothesis that this drug activates or this drug inhibits a target and modulates the disease in a particular way in a particular patient population can be turned out wrong if you do it in the later stages. So another question that can be put up from a lot of people is that how can we identify which proteins are like deregulated in a disease condition and which are needed to influence to achieve cure. So proteins come from gene, so it is easier to study genes. Okay, to answer this question, it is easier to study genes than to study proteins. So one approach to finding new drug targets involve comparing genes, because it's easier to study genes of healthy individuals with those of people with disease. So we compare the genes of a healthy individual, healthy person, and we compare it to the genes of a disease person or unhealthy patient, we can say, or a patient for that matter. So the difference between these two genetic maps, whatever the difference we'll observe in these two genetic maps, can help us to generate hypothesis on which protein, okay, first of all on which protein to act, and second of all or which protein is lacking because of which this disease is occurring, and we can because of which the disease is caused. So it is possible to do the opposite, that the another approach based on gene can be that first of all we compare gene, second of all is we can do is to do the opposite, that we can change one of the genes. We can change one gene at a time in a cell or like simple organism and observe the resulting effect, whatever is the resulting effects, the like so-called phenotypes of the mutation. So in a way we are also doing a mutation, and we observe whatever the changes are coming, whatever is the aftermaths. If the phenotype have some similarity with the disease state, it can give ideas about the possible relation between the mutated gene and the disease. So if it is similar to a disease condition, then you can sort of provide a relation between a mutated gene, and you can provide a relation between a disease condition, that because of this particular. Another approach is to start already with the bioactive substance, for example a natural medicine using in traditional medicine, or a compound from a basic research or known drugs with unexpected effect. So that is what we discussed here, like this is what we are talking about, target deconvolution, that we actually in a sense they are talking about it, that we take a drug that we already know, it can be an active substance or it can be a natural medicine or a traditional medicine for that matter, and we take it and then we look for some unexpected effects, whether this particular active substance or known drug or this particular traditional medicine has an effect on our disease condition. So we'll look for the unknown effects. मतलब हमें कुछ effects तो उसके पता होंगे, but अब हम उसके unknown effects को देखेंगे. For example, aspirin, the active components of the willow bark extract is used to cure fever for a long time. If we are taking the example of aspirin here, so its target protein would obviously be cyclooxygenase if it is used in the treatment of fever. So its target protein cyclooxygenase is also the target of modern anti-inflammatory and analgesic drug. Here aspirin, which was used traditionally from like willow bark trees, would act on cyclooxygenase to treat the fever, but cyclooxygenase is also used for modern day drugs, also used by modern day drugs for like NSAIDs, that is anti-inflammatory and analgesic effect. So there is something known as chemoproteomics. Chemoproteomics, and it is not easy to find out which protein binds to a chemical substance. So a rich discipline called chemoproteomic offers a wide range of methods to study just that, and we call that as chemoproteomics. Now, genomics in target identification, that is also something very popular nowadays. So it could be asked, genomics in target identification. So there are many things like genomics, bioinformatics, proteomics in target identification, and it is like a very interesting and good topic in itself, which we will study in the next sub part, because already this video I think is going to be very big if we included all these topics. So we'll study it in part 2, subpart 3 of drug discovery and development lecture series. So make sure to watch this part for if you want to learn about genomics and drug target identification, bioinformatics and proteomics. Very interesting, very hype topics nowadays. Till then, like, share and subscribe, and turn on the post notification if you want to get daily updates regarding the topics. Bye.